Good day, this is Professor Will, CBMD, PhD. And today is Tuesday, November 30th, 2021. It is 9:35 and 46 seconds, p.m. Eastern Standard Time. Today's research, CRISPR. So let's hop to it here. The scientists produced new antibiotics by gene editing. Date November 29th, 2021. Source, University of Manchester. The scientists have discovered a new route to produce complex antibiotics, exploiting gene editing to reprogram pathways to future medicines, urgently required to combat antimicrobial resistance, treat neglected diseases, and prevent future pandemics. Researchers from the University of Manchester have discovered a new way of manipulating key assembly line enzymes in bacteria, which can pave the way for a new generation of antibiotic treatments. Researchers describe how CRISPR-Cas9 gene editing can be used to create new non-ribosomal peptide synthetase, NRPS, enzymes that deliver clinically important antibiotics. The NRPS enzymes are prolific producers of natural antibiotics, such as penicillin. However, up until now, manipulating these complex enzymes to produce new and more effective antibiotics has been a major challenge. The UK government suggests antimicrobial resistance, AMR, infections are estimated to cause 700,000 deaths each year globally and are predicted to rise to 10 million, costing the global economy $100 trillion by 2050. AMR also threatens many of the UN Sustainable Development Goals, SDGs, with an extra 28 million people that can be forced into extreme poverty by 2050 unless AMR is contained. The Manchester team says the gene editing process could be used to produce improved antibiotics and possibly lead to the development of new treatments, helping in the fight against drug-resistant pathogens and illnesses in the future. The emergence of antibiotic-resistant pathogens is one of the biggest threats we face today. The gene editing approach researchers developed is a very efficient and rapid way to engineer complex assembly line enzymes that can produce new antibiotic structures with potentially improved properties. Microorganisms in our environment, such as soil-dwelling bacteria, have evolved non-ribosomal peptide synthetase, enzymes, NRPS, that assemble building blocks called amino acids into peptide products, which often have very potent antibiotic activity. Many of the most therapeutically important antibiotics used in the clinic today are derived from these NRPS enzymes, e.g. penicillin, vancomycin, and daptomycin. Unfortunately, deadly pathogens are emerging, which are resistant to all these existing antibiotic drugs. One solution could be to create new antibiotics with improved properties that can evade the resistance mechanism of the pathogens. However, the non-ribosomal peptide antibiotics are very complex structures, which are difficult and expensive to produce by normal chemical methods. And to address this, the Manchester team used gene editing to engineer the NRPS enzymes, swapping domains that recognizes different amino acid building, leading to new assembly lines that can deliver new peptide products. Researchers added they are now able to use gene editing to introduce targeted changes to complex NRPS enzymes, introducing alternative amino acid precursors into peptide structures. Researchers are optimistic that their new approach can lead to new ways of making improved antibiotics, which are urgently needed to combat emerging drug-resistant pathogens. Good day, this is Professor Will, CBMD, PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.